So just to illustrate some of the points from the last section, here's some little pictures. Look at them. Does this make sense? So apomorphies, what are those? You remember? Something that's different from the original. So if the original is white dot, then if you're still, in this new time, the same as the ancestor, if you're still white dot, then that's a plesiomorphy. And if you have changed into black dot, then that is an apomorphy in the simplest possible term. Yeah, and most abstract. Autapomorphy, remember, is a unique difference which is not found anywhere. So it's derived, but it's not shared. A synapomorphy is derived and shared among a group. And so a synapomorphy is wonderful for defining particular groups. You can say all crocodiles have various features. And then homoplasy. Homoplasy is the enemy. So homoplasy is when you end up with the same character, but potentially by different means. So you reconstruct the ancestor as white dot. Most of the intermediate relatives are white dot, and yet here's black dot and here's black dot. There are different ways this can happen, but a big one is going to be convergent evolution. If there's some new change or some particular environment where black dot is the best dot to have, then that's what you got to have. Yeah. So, and it'll be selected on multiple branches, particularly if it's a small change. Generally, you get these homoplasies in simple character states like a nucleotide or an amino acid. Generally, you would not expect to find them at the level of like an entire gene or multiple genes, except if it happens by horizontal gene transfer. So vertical gene transfer, that one that's like, your grandma to your mom, to you, to your kids. Yeah. Transmission of genes through direct descent. Yeah. Horizontal gene transfer is like if you were transferring genes to the genome of somebody else that's alive today. Or if we took some of your DNA out and cloned it into a potato, just because. Yeah. I don't know why. We got a grant to do that, I guess. Yeah, that would be horizontal gene transfer because the potato is not your son. Your little round son is just a potato. Yeah. All right. So synapomorphies, once again, so you can see the red dots, red squares are a synapomorphy. The orange squares are a synapomorphy. And these are the ancestral state back here. So there you go. Two populations that share a common character state. Okay, fine. A couple more definitions, and then we'll actually get on to the tree building stuff. So when we make these trees, they can be good or bad, basically. What we want is a monophyletic tree, which means that all of the little branches here share a common ancestor. Now that's never directly provable or testable unless you have a time machine and probably a microscope and a molecular biology lab and some really good timing. Yeah, and then you need all the intermediate samples. No, that's terrible. Yeah. But it can be inferred with a certain statistical confidence. And when the confidence is high enough, we say, you know what, that's a reasonable inference based on the data that we have. And if you get more data, the inference may change because more data is better, and it's the law of large numbers. Basically, if you take more coin flips, you're going to end up closer to the actual difference between heads and tails. Any anomalies will tend to even out over very large numbers, essentially. So yeah, monophyletic, good. Non-monophyletic, bad. You can break that down into two different groups, which are paraphyletic, where you take some but not all of the things in a group. So like if I was to say, all right, the halophiles, they're one thing, and all the rest of these guys, we're just going to call them, well, here, these are all methane-producing archaea. And so those are paraphyletic because the halophiles are within this. They're a descendant from a common ancestor that gave us all of these things. So methane respiring bacteria would not be, or archaea would not be a proper monophyletic group. They would be paraphyletic. Polyphyletic would be to take things that are similar in some apparent character but do not share a common ancestor. So, like if you say slime molds are kind of slimy and they just have one cell, and you say, well, those are probably closer to bacteria, let's say, which can be slimy and are, live one cell at a time. Except that slime molds are weird. They do a lot of other things. Yeah. Anyway, that would be an example of a very bad assumption because we would be drawing a circle around this and this, but leaving out all of this and all of this. And as you can see from the branching pattern, we're leaving out a lot of ancestors. That's the thing with phylogeny. You're not allowed to leave out any ancestors. All the ancestors got to be there. All the grandmas, all the grandpas, all the aunts and uncles. It's a enforced family reunion. Yeah. Okay. Example in a picture, just because it's nice to have a picture. So 1 and 2 would be monophyletic. 1, 2, and 3, you could draw a circle around that. That's monophyletic. 4 and 5, monophyletic. The whole thing, monophyletic. What would be an example of a paraphyletic grouping? You say like 2 and 3? Yeah. 3 and 4? Sure. What would be a polyphyletic grouping? Like 5 and 1, but not 2, 3, and 4? Sure, why not? Yeah, and those would both be bad. Nobody ever sets out to make a paraphyletic or polyphyletic reconstruction. But when you find problems in reconstructions, those problems have these two words. So like the problem is that we have a paraphyletic reconstruction at this character or in this group, or we have a polyphyletic one. Yeah. It's just our word for describing when things go wrong. This is how it went wrong. Okay, almost there. So homology is common descent. It's a shared common ancestor. And so this can be for a gene. It means there was originally one gene that got, somehow copied inside an organism or horizontally transferred umpteen times, but they do definitely share a common ancestor beyond a statistical reasonable doubt. Yeah, because it's inference. All right. However, if that character is evolved independently, then that is a homoplasy. Yeah. So homoplasy looks like homology, but is not. And so while one character may be a homoplasy, generally you can tell that that one is not representative of the entire evolutionary path, because you can look at all the other stuff around it. So context is really important in deciding if you're dealing with homology, homoplasy, or just two things that really are not comparable and should not be on the same tree in any way. So yeah, you can have loss of features, which would lead to a reversion of an ancestral condition if you had a new feature that was evolved that was like an extra gene and then you lost it. Think like blind cave fish, evolved from fish with eyes, like normal fish, but yeah. It got broken one day and the lights were out and nobody noticed and everything was fine. And so, yeah, it just happened and now they're eyeless, like they're super, super, super ancient ancestors. Parallel evolution, convergent evolution, yeah, when like the environment is selecting something or it just happens independently. All right, so yeah, polyphyly happens all the time, and yeah, you know what? That's all we're going to say about this. We're going to skip over those two. Okay, so reconstructing or constructing phylogenetic trees, really attempting to reconstruct the actual phylogeny in the form of a constructed phylogenetic tree. That would be a more precise way to say it. So yeah, we're going to use homologous characters as far as possible, and that can be like an entire protein with a bunch of little smaller characters inside it that we know together make up a homologous pair. And we will use this to reconstruct the evolutionary path. So That's coming up in the next section. Stay tuned.